me. That's perfect. Share my screen, share my screen. Perfect, okay. So if I call on you and you're discussing with me at the level that we've been discussing the topic and what was expected of you from making your concept maps before coming to class, then you'll get full credit. It doesn't matter if you're right, but if you're in the realm of what we're talking about, I'm going to give you full credit, okay? But if you sit there and you say, I don't know, then I'm not going to give you full credit. You'll get participation points. So you'll always get some points just for having showed up. There is opportunity for making up those points too. So if you come in, you're like, I want to be able to answer this question, but I just don't know it. There is a chance for you to make up those points later. So you can always email me and say, this was the question you asked me. I didn't know the answer, but this is what the answer is and why. And then I can give you the full credit for those points. But that's on you if that happens, okay? We clear? So that's on you if you want to make those points up or not, all right? I'm not going to email you and be like, you got partial credit. Do you want to make up those points? You're adults and you can reach out to me with that stuff if you want to get to the full points. So there is opportunity if I give you partial points, and I will put the points in within usually 24 hours, but give me up till the end of the week, say Friday. And so if you notice that you got a partial credit on something, then you can email me and be like, oh, I forgot about this. Yeah, I want to get full credit on that. So we're going to go over like all the assignments and everything. So before before we go into that, I want hands raised. How many of you guys are going into nursing? Hands raised. You're planning on going into nursing. Okay. How many of you guys are going into vet tech? Hands raised. All right. Anybody whose hand didn't raise in one of those 2 times? That's everybody? Okay. So in this class, very, very clear here. Everybody in this class is going into healthcare in some form, whether human or animal. Yes. How many times do you expect to see your patients, human or animal, to be behaving exactly according to how a textbook told you? Right, and that's wild, right? To even think that, right? That is why we have to get away from flashcards, Quizlet memorization, and regurgitation, right? We have to understand the concepts behind it so we can problem solve. So if you're the nurse on the floor taking care of somebody that got out of surgery and they're having X, Y, Z symptom, you know when it's appropriate to call for the doctor, or you know when it's appropriate to ask the charge nurse for help or whatever, right? Because you can problem solve and you can know better what this scenario is applied to that patient or that situation, whether it is in vet or nursing. Now, I talk a lot about nursing in this class. If you're going into vet tech, the concepts still apply to you, right? But you'll just have to think about it in the context of the animal. But majority of my students are going into nursing. So most of the way that I'll talk about things is focused on human medicine, but we'll talk about animal medicine too. We'll talk quite a lot about rabies. And if you are undereducated on rabies, hopefully this class will help you understand a little bit the dangers associated with rabies. Rabies is a 100% fatal disease. I say 100% and I quite literally mean it. If you have any signs or symptoms having rabies, like let's say you got bit by a raccoon, and a month later you start, I feel sick, you're done, you're out. 100% you will die. 100% you will die. You won't make it. There's nothing medicine can do for you at that point. But we'll talk about post-exposure prophylaxis, which is like giving you the shot as soon as you got bit, giving you immunoglobulin when you get bit, and things like that that can protect you from getting to the symptom stage. But once you get to the symptom stage, there's literally nothing we can do for you. So we'll talk about that stuff in this class to put things like that into perspective. A lot of what we're talking about applies to diseases, so infectious diseases. So we also we'll learn in the class, hopefully though, we have this general concept of, when you come into this class, people automatically think of infectious diseases. But then I ask you to name me one disease that isn't infectious based. And when we're in the classroom, people's minds go blank. And we got to get away from that too, right? Don't just think about the situation that you're put in that moment, because your patients might have other things going on. They had surgery for their knee, but they might be having signs or symptoms associated with their heart disease, and you're thinking about their knee. But we got to get away from that too. So heart disease, for example, not an infectious disease, but it is a disease. Stroke, diabetes, many types of cancer, most types of cancer are not associated with infectious stuff, right? So those are all disease. Disease is deviation from health. So we're not talking about diabetes and stuff like that in this class. We're talking about the infectious diseases in this class. So we've got to understand our enemy in order to fight him. And that's what this class is about, understanding our enemy, okay? So that makes sense why we're going to be doing the application stuff. So it'll make sense what we're doing with that and what I expect of you guys when I call on you, but that's why we have to apply. We can't just memorize. It's not enough for you guys to say, bacteria have a cell wall of peptidoglycan. Well, what good is that going to do you in the healthcare field? Just knowing that fact. And it's not. If you just have that fact in your brain, and we all know, how many times have you guys studied for a test? Maybe passed the test and did just fine on it, but then forgot that information a week later. That's not going to serve you when you leave this class, right? Like if you're going into medicine, you're going to be seeing people with infectious diseases. I need you to have context. I don't necessarily need you to memorize the steps of the gram stain. That's not going to serve you in the healthcare field. Maybe if you're a vet tech, you'll actually be doing gram stains and stuff like that. But my nursing students won't be. They have the lab for that. So it's not going to serve you necessarily to know you put 5 drops of crystal violet. But if you have a context of why we do the gram stain, then that gets you better understanding and better understanding like what your results are from your patient, why we're doing a gram stain on their CSF, and how that relates to medicine practice. Okay, so the concepts are way more important in my course. I do require that you guys have taken chemistry before you come here. That's actually a state requirement. It's not my requirement. So if you take microbiology, you have to have had chemistry. And most people come in here and they have dumped their chemistry from their brain. I hate to break it to you guys. We're going to be using chemistry every single day, even probably today, but not the math, not balancing equations. I could care less about that stuff, okay? Like that stuff, I'm just whatever. Good on you if you were good at the math part of chemistry, honestly. I have my bachelor's is in biochemistry and I still don't, I'm not good. I go within like the SSOC to try to cover for when the guy left from the SSOC in the STEM lab. And I was like, people come with me chemistry. I have to convert the molar mass. And I'm like, cool. I don't know. So, but the concepts behind that stuff, what an acid is and what's going on in a solution when it's an acid, pH and why it's important, hypotonic, hypertonic, all of those things. We're going to be talking about that stuff every single day. I cannot tell you how important pH is going to be in biology without you actually like getting involved and seeing how cells work and all that stuff. But hopefully when you do chapter 2, which we'll be going over next week, right? That's week 2 thing. That'll be your chemistry review, and it reviews the stuff that's going to be important for this class. So understanding what a covalent bond is and what's going on with the electrons in a covalent bond. Their atoms are sharing electrons in a covalent bond versus ionic when they're donating your electron, versus a hydrogen bond, versus what's going on in low pH versus high pH and all sorts of stuff. And probably the most important bond, well, they're all important, right? But when your proteins have functions in your cell, and like, let me make this clear too. You have 20,000 genes in your genome as a human being, okay? 20,000. All of those genes are either a gene to code for a protein or genes that regulate those. So we're talking genes for RNA, like ribosomal RNA and tRNA, that help build the proteins, or that it's a protein or something that turns on or off expressing the protein. You don't have genes that make fat. You don't have genes that make carbohydrates. You don't have genes for those things. You have genes for proteins. So literally every single thing in your cell is governed by a protein. A protein is made that helps make fats. A protein is made that helps build carbohydrates or break them down and stuff like that. But the proteins govern everything. And the proteins, their shape is way more important than their literal amino acid sequences. And so how their shape is defined is usually defined by things like hydrogen bonds and how the R groups, you guys learned about this stuff in chemistry. Is this at least sounding somewhat familiar to you guys, amino acids and the R groups? So how those R groups, and don't worry, chapter 2 reviews this stuff, okay? But how they interact with one another determines the shape of your protein. And they often interact because of charges. Well, you know, very basically stated, but we call them dipole moments, right? The H's have tendency to be a positive dipole. And the oxygens tend to hog electrons, so they tend to be negative. And so negative oxygens like to hang out with positive hydrogens, and those are called hydrogen bonds, those attractions that develop there. Hydrogen bonds are just attractions between a hydrogen that has positive charge. So proteins are folded because of that. So whenever you have pH shifts, a pH that's low, an acid, means you have too many H pluses in the solution, more H pluses than you usually have. And so if your body is supposed to be between 7.35 and 7.45, and that's quite a strict pH range, but that's what your body is supposed to be in for a reason, because your proteins can't fold properly if there's too many hydrogen ions interfering. Your proteins can't fold properly if there's too few hydrogen ions in the solution. And so that is what we're talking about with the body's pH. And when somebody goes acidotic and their pH falls, that's why that's important. Because now too many protons in the body means their proteins can't fold and function properly. That's big picture stuff, right? That's looking at not just knowing what pH is, which is a little niche thing, but seeing how it applies to a big system. That's the goal in my course, to get us thinking at that level. Another thing that kind of puts things into perspective for people. When you guys feel sick and you feel like crap, right? You feel like, we call it like social avoidance. When you're sick, you don't want to go out and hang out with people, right? You're too tired, you feel like junk. You just like literally, even if there was a cool thing going on, you don't want to go be around people. You feel pain, that keeps you at home. You feel tired, that keeps you at home. You feel like you don't want to eat sometimes, right? So all these things that we think of as just, oh, my immune system is beating me up, this sucks and I can't live life. Did it ever occur to you that like there's an evolutionary advantage to keeping you at home instead of interacting with people when you're sick? There's an evolutionary advantage. Your body, that does you good and it does the community good to make you feel like crap and stay at home. That's big picture thinking though. Yeah, you feel like crap when you're sick. There's a reason. Not the reason being, of course, your immune system made you feel like crap. That's why you feel like crap, but there's a reason to feel like crap. Keeps you away from people getting other people sick. Keeps you at home so that your body can rest and recover. Keeps you from eating things so that your GI system can reset and recover. There's reasons for all of these things that are actually advantageous. So we're going to talk about stuff like that. We want to get outside of the box of saying 98.6 is a normal body temperature. Go meet with somebody that knows old people and ask them if 98.6 is the normal body temperature. Because if an old person, I'm saying old, like somebody who's 80, 90 years old, if their temperature is 98.6, that's a fever. Yep, that's a fever for an old person. Get out of the box. We got to get out of the box. And so that's what the point is of this lecture course. Our in-person part of the course is going to be us getting out of those boxes wherever we can. We got to start thinking that way because the nursing program, they're going to want you out of those boxes. They're going to give you scenarios in their NCLEX questions where every question is right. But you have to know which one is the right one for this scenario. The most right. And so we've got to get into that thinking. So that those of you who are going to nursing school, you can think outside the box. If you're going to vet school, that type of thinking is going to help you too. But we just got a particular issue with this in nursing school that we know we can target there. Half of the people that get into the nursing program won't make it through. I want that number to change. I want it to be all of you guys. So I'm trying to prep you guys here so that when you get into nursing, you're not dropping after your first semester and you're not having wasted your money getting all your prereqs done and then not make it into nursing school. And then what do you do? So I don't want that to happen to you. So we're going to raise that number, right? And you guys are going to be part of that. I'm not going to make you guys raise your hand or anything, but I'm sure some of us heard about how there was somebody that worked in the STEM lab in the SSOC, which is the Student Success and Opportunity Center, where they provide tutoring and things like that for students, that we had a STEM lab tutor that was handing out answers to the exams for all of the STEM classes. I mean, quite literally all of them. And I don't mean like, here are some answers. I mean, it was literally the exam questions verbatim and the answer to what that exam question was. And I changed my exams every semester and he would hand out a new one every semester. I didn't know this until somebody shared a Kahoot practice quiz with me. And they were like, can you look over my Kahoot and make sure that everything looks correct? And all of their Kahoot was my exam questions. And so I kindly asked the student, I had no reason, you're sharing that with me. I'm sure you know that. You don't know that that's my test, right? Like there's no way that somebody would know it was my test and then be like, can you check that this is your test? So I contacted her and she said she got a handout from the STEM lab person. And so that's how that got caught. I can't speak for every other professor, but when I found that out, which was halfway through last semester, halfway through the spring semester, I rewrote all of my tests that second. So all of my tests have been rewritten. So if you have those study sheets from the STEM lab, you can use them to study with. I still test the same concepts, but the answers and questions won't be the same. I'm just letting you know that so that we're transparent there. I can't lean on that being the answers to my test, but the concepts are still the same. Yeah. All right. So let's go through how the course is set up, and we'll go over the lecture stuff, and then we'll go over the lab stuff during the lab time. Sound good? Okay, and today we will be jumping into calling on you guys already. Now I know for a fact that you guys haven't read chapters 1 and 3. That's okay, don't stress. Just do your best to guess at the answers when I call on you. And if you're participating, you'll get full credit just for participating today. Okay? So we're not going to be judgy. And I will, of course, walk you through everything. Deep breaths. Okay. So, all right. So this is our class. Sorry, I tried to set all of my classes up as like a fall color theme. This is our class. And when you get to the first page, my home page is my syllabus. So that way it's always there whenever you first come in. At the top, you'll have your first 3 announcements, whatever the most recent ones are. If you open them, if you actually open my announcements, you read them in the emails, that's fine, because you guys should have your Canvas set up where you get emails if I post announcements. If you haven't gotten any emails from me yet, you need to change your settings on Canvas because I've already posted a couple of announcements, right? So make sure that you are getting emails from my announcements. Any changes in my course, I put on the announcements. I don't send a separate email. So just make sure that the announcements are there. Or like if there's a weather event, check the announcements and see if I posted anything about that. If the campus closes for weather, because towards the end of the semester, we might see weather. And we, you know how it is in Oklahoma, sometimes we get tornadoes in the fall too. So if there's a weather event or something else that causes the campus to close, generally speaking, if there was a lecture that day, I'll have an online version of the lecture for you. If there was a, or I'll have you guys like answer the activity stuff on your own time. Just depends. And I'll let you guys, I'll give an announcement telling you guys exactly what from you guys. So yeah, things like that, lab stuff, if there's a weather day for those, I usually try to combine the missed lab with another lab, but if we can't do that, then I'll just cancel it and you don't have to know it, like quite literally. So that usually only happens if I have multiple cancellations, like multiple labs canceled in a row. So don't bank on that, okay? But I'll have all of any changes to the course. If I have a doctor's appointment or I have an emergency or whatever, anything that you guys need to do in relation to that, I post it in the announcements. Okay. I have something called blunted cardiovascular response, which is something where my body doesn't react to stress the way that it's supposed to. You could literally get in a car wreck with me. You could scare me. You could like, I could wake up in the night and you'd be hovering over me with a knife trying to stab me or something. And my pulse rate doesn't go up. It stays 60. And my blood pressure doesn't go up. It stays 100/60. And so I don't have a stress response to emotional stresses, but if I go run, watch my pulse go up. It's normal. So, yeah, right. So I don't know. And so some people are like, wow, so you don't get stressed out. No. Everything else, like mental stress, and I'll get chest pain from anxiety and things like that. And I actually get chest pain because my heart rate doesn't go up when I'm having all those other mental effects of my stress. So I have that, and it causes my parasympathetic, if you guys have taken physio, you know what I'm talking about, but my parasympathetic response, which is your recovery response, doesn't regulate properly. And so that is parasympathetic dysregulation. And so the reason I'm bringing this up is my parasympathetic system, because it's dysregulated, it's delayed and overreactive. So if I have a stressful event and then I relax at home, it takes like a day for my body to be like, oh, we relaxed. Oh yeah, sorry, here, relax. And it like screams it out. And what happens is all of your parasympathetic responses, not just the emotional ones, but your physical ones as well, like your digestive enzymes and other things that are controlled by your parasympathetic stuff, goes haywire. And so that leads to me having pancreatitis. And so it's a weird way of me having those symptoms. I don't know if you guys know this, if you've worked in hospitals at all or had people that have had pancreatitis, it's just extremely painful. And I mean, we're talking level 10 blackout painful. So when I have those attacks, if I have them during class, which it happens sometimes, I try to push through it, which is why I have the stress problem to begin with. But I try to push through it. But there might be days where like I'll have to take a break or something like that because I can't handle the pain. I mean, I have blacked out from the pain before. So it's it's not a joke. It's not something that's minor. And of course, you know, to manage this aspect of how my body handles stress, what do the doctors say? Reduce your stress. And I'm like, I work out, I do yoga. I mean, I'm overweight, but we're working on that, right? I eat healthy, and I've been doing everything I can since I found this out, but it takes a lot of time to retrain your nervous system. So bear with me, please. But that's why I bring up the medical stuff, just in case something like that happens. It shouldn't be an issue, maybe once a semester. I haven't had any issues with it like frequently. So just bear with me with that. I am a doer and I like to be in control of things. And so whenever I start having to relinquish control, that actually causes me more stress. So I have to find a way to balance that. That's on me. But anyways, here's your announcements. This is your syllabus page. on the syllabus page, whether you access it from home or from syllabus, it shouldn't matter. You'll see this jump to today button. If you click on the jump to today button, it'll usually jump to today and then scroll back up. That's okay, just scroll back down to it. And this has all the assignments that are due today or whatever the next day is that you have something due. So you guys have stuff due on Thursday. And every week I will send an announcement letting you guys know what is due when also. So yes, you can access your assignments in Student View on assignments. Your assignments will be organized. Let's do it so we can show you so that I'm not just talking about a thing. You can actually see the thing. The internet is really slow in this building and I apologize. So, all right. So when you guys access the assignments on Student View, it'll first show up as like the different types of assignments, but then it should resort once it finishes loading into upcoming assignments, show by date. You can also show by type, but if you show by date, like it's set up here, it just is in order of when these things are going to be due and what's due and whatever. So you can click through that way, or you can go to the syllabus and do the jump to today thing. Or I will also be sending emails out. So you'll get an email today, I think, reminding you, or today or tomorrow, reminding you that you have stuff due before lab for Thursday. And this is what you need to get done by lab time. Okay, so have that email. But then every Friday at 8 a.m., you'll have this is what's due by Sunday at midnight, and this is what's due before my next Friday update and what day it's due and everything. So I will send emails with that. And those also include what learning objectives you were supposed to have learned that week for the lecture stuff. So there's really no reason to get behind on the work because there's always going to be reminders, either in your to-do list, on the assignments, on the jump to today, or from the emails that I'm sending you guys to remind you to keep up with that work. Okay. So that is there on that syllabus page, and syllabus is here as well as in the link on that first up module. Same syllabus. It has my office hours on there as well as in the instructor area, which talks a little bit about me. A little bit about me, I got my bachelor's at OSU Stillwater in 2006 in biochemistry. I graduated magna cum laude from OSU Stillwater. And then I went to my graduate work. I was getting a PhD at the University of Pennsylvania, which is an Ivy League school. And so I was doing my graduate work at an Ivy League school. I was collaborating with the CDC and the Army on my research. I was giving talks around the nation and around the world. I was doing international talks as well. And then I got married. And my husband found his dream job at Tinker Air Force Base. And he said, I'm absolutely never moving up to the Northeast because I was going to school in Philadelphia. I'm not moving up there. It looks like you wanted to work at the CDC in Atlanta or with the Army in Maryland. Yeah, that's not happening. So we got married, and then within a month of us being married, he's had this ultimatum that, yeah, no, we're not doing that. And so I quit graduate school for him at 24 years old. I was 24, so to put that in perspective. And it was a trend with that marriage. So that's my first husband. We got a new husband now because that ended up being a trend, and I've grown as an adult and realized that my value is the same as everybody else's, just like yours is. Okay. So I don't have a PhD. I quit when I was halfway through that program. I have a master's degree, what they call a terminal master's degree. I did well more than what a master's degree requires, but they couldn't give me the PhD. So, and I never went back to finish it because once I moved back to Oklahoma, my husband was like, well, you need to get a job. And so I had several jobs in industry. I've done quite a lot, had years of work of phlebotomy experience. So if any of you guys are ever working on your sticks or want advice on phlebotomy stuff, I can help you with that. Whether it's animal or human, the principles are the same. I'm a very good stick. And I ran the platelet machines for OBI for a year. And that requires a larger needle than what you typically use, like with butterfly needles. 12 gauge for the, not 12, excuse me, 20, I don't think it's 20, I think it's 18. 18 for the platelet machines. We use 14 gauge for regular blood draws. They call them 16, but that's the bore. The actual needle is 14. And if you've seen the needles, man, if you can stick with those needles, you can stick. And those needles have to stay in for 10 minutes drawing blood, not for a couple of tubes, right? And so I'm very good with phlebotomy. I did 3 years at OBI and then 2 years with DLO. So we're talking about sick patients with DLO, right? And I did do butterflies and vacutainer needles with DLO. So I'm very good if you need advice with that. After that, I did the HLA lab at Baptist, which is their transplant immunology lab. So I did all the work for determining if patients would get organs or not, or if they were likely to reject them or not, doing all that blood work for that stuff on the immunology side. That was a very stressful job. A lot of on-call work. You get called at 3 a.m. because somebody has a donor kidney coming and you have to test it before they can do the surgery. And then the patient and the surgeon are waiting for your call. And that was a little bit much for me. You know, kudos to all of you going into healthcare where there's lives that are actually going to be depending on your work, because that was too much stress for me. I couldn't take it. So I went into food safety testing after that and did a lot of testing for local companies, including McDonald's and stuff like that here in Oklahoma. So I've experienced working in industry. I've experienced working hourly jobs. And then I found this job, which I love. Teaching is my passion. I love teaching and sharing with you guys the understanding of science. That's my real passion. So I love it. And I'm happy to be very good at that as well. My first year eligible won a teaching award, the highest award you can get at OSU. And first year eligible, I won that award. Nominated by students, to be clear, not by my fellow faculty, nominated by students. And I got more nominations from students than any other professors ever had. So hopefully you guys will agree with that. But yeah, so I have that award and I hold that very close to my heart. And I work every day to be better. Even though I got the award, that's not it for me, right? I want to always be better even than that. And so that is important to me, to be always advancing. So that's the type of teacher that you have. And I say that I studied at an Ivy League school for graduate school. The professor that I learned advanced immunology under, his name is Drew Weissman. He's the guy that developed the mRNA vaccines for COVID and won the Nobel Prize for that. So that's who I studied immunology with, to put that in perspective. So I'm very good with that stuff. And if you guys wanted to get into vaccine discussions with me and how vaccines work and why the mRNA vaccines can't become part of your DNA, they can't. Like it's like if you turned into a frog, it's that level of you can't do it. Then yeah, so I'll be happy to have those discussions with you. And we will be learning vaccines this course. We do teach vaccines as though vaccines are a good thing for you to be doing. Now, I want to be very clear about this. Are there risks with vaccines? Let's be real here. Are there risks with vaccines? Of course there are. But I need you and your family members to understand. When we talk about vaccines, we are not talking about getting a vaccine versus not getting a vaccine and living your life having the vaccine or not having the vaccine. When we get vaccines, we're talking about the vaccine versus the disease that's associated with it. So one that I want to put into perspective, of course, is measles. Right? Measles vaccine is hands down the most effective vaccine that we've ever designed. Hands down. You can get measles vaccines when you're young as a kid and have protection for your entire life. And I mean total protection, not like COVID vaccine where your body's trained to handle the virus like a virus instead of freaking out from it, which is how the COVID vaccine works. Okay. But with the measles vaccine, it actually keeps you from getting the disease. Now, if you get measles, the odds of you just having a rash and recovering from it is pretty high. You're probably just going to get a rash and recover from it, most people. Yeah. But with the measles illness, you're actually not likely to develop immunity from having the illness. The way the actual live virus works, you won't develop immunity from having the live virus. Most people won't. So you can just get it over and over and over again. Each time that you get measles, you're even more likely to develop meningitis or encephalitis. Okay. So now I want to put that into perspective. Encephalitis, not a joke, not a fun thing to have. And we're talking about little kids, yes? So as it turns out, the mortality rate in unvaccinated people with measles, the mortality rate, we're talking people that die from measles, is 1 in 1,000. And 9 out of 10 people that you come into contact with that's not vaccinated will get the measles. It's the most contagious disease on the planet. It's more contagious than the flu, the cold, or COVID. It's just most people are vaccinated right now. So that's why we don't get it as much. But the more we get away from vaccination, the more people get the measles and the more people are going to be dying from measles. 1 in 1,000 die. So keep that perspective in mind. That's what we're looking at when we talk about vaccines. We're talking about people dying. We're not talking about people having, 1 in 1,000,000 might have a side effect. 1 in 1,000 will die from this. Right? So this is what we're putting in perspective here. If nobody was vaccinated, 90% of the population would get measles easily, probably 100% eventually. Nobody was vaccinated, and 1 in 1,000 of those people would die from it. That's what the measles really is. And so we've got to put that into perspective when we're talking about vaccine injury. Now, We all are aware that there is absolutely no link between the measles vaccine, the MMR vaccine, and autism. We're aware that the actual science, there's no science that supports that. This is a fact. There was a paper that was published that was completely falsified. It was paid to publish that data, and it has been completely retracted because it was all falsified. And the doctor that published that paper lost his medical license in the country that he practiced in, which was in the UK, and now works under RFK. That's all I'm going to say. That's all I'm going to say. Okay, so just stay educated on this stuff and know your facts. Have opinions, have them. If you don't want to get vaccines, I'm not going to stop you. But if your laws prohibit me from having my insurance pay for my vaccines and for my children's vaccines, I am going to come for you. Yeah. So you don't want to get vaccines. You don't want your kids to get vaccines. That's fine. That's your choice. But if you take away my rights, we got a problem. Okay. Education about facts, not opinions here. I want to hear it. And if you guys want to have the personal discussions about it, please come see me because I'm happy to do it. But understand where my background is and where my opinions come from, my facts, not from the people telling me this. Okay, not from Instagram. All right, or TikTok. I don't even get on TikTok. I can't with it. All right. So, anyways, so you guys see how you can navigate the course a little bit with the stuff that is due. The other side that you can navigate the course from is modules. If you're a modules person, okay. So I tried to set this up as clearly as possible with this so that it was clear what is what and how it's organized. Once you get out of the first up and the note-taking modules, my weekly modules are all set up the exact same. So it should be pretty easy to navigate. We have the first up module, and in student view, you will see that it asks you to complete certain things to open the rest of the stuff for the course. So you have to do anything that has a little circle next to it has to be turned to a check mark. I haven't done it for this course yet, but basically, like, for example, the lab safety document, you just have to open this. So I'm opening it right now. Oh, cool. I didn't even click on the link. I haven't even looked at the document yet, but I opened that, right? So I can go back to that module, refresh it. Hold on, give me a second. The internet. And now I have a check mark there. You see the green check mark? Okay. So some of those you just have to open it. But the only one that's going to be a pain is this one down here. It's the test your computer with lockdown browser. Make sure that your lockdown browser works on your laptop or your computer that you'll be taking the test on in class. My tests are in person. I have zero respect for online test taking. Zero. And so it's not that I don't trust you as an individual. I'm sure you're wonderful and you're honest. But there are plenty of people that aren't. And it's actually pretty easy to get around those things if you know how. I'm just saying. So I don't have respect for ProctorU, for sure. Everybody hates ProctorU to begin with, but I also don't have respect for online test taking. I know Dr. Shearer is a big fan of, but he uses the lockdown one. And it's supposed to flag when people are looking away, and it records you the whole time. And so he'll actually kind of review those if somebody got a certain score and see does their behavior, like if they got 100 on the test, we're gonna watch your video and make sure that you're not looking at something incorrectly. But the issue is what we've had in the past is you can actually have a computer that somebody else has also on, and you're looking at one monitor, and then there's a second monitor, and they're mirroring each other, but the camera is facing this way, and somebody else is taking the test. So you're just sitting here reading through, looking like you're clicking on stuff, but somebody else is taking your test. And so we actually caught that because the person that was taking the test on the camera fell asleep, but somebody kept taking the test. Yeah, somebody kept taking the test behind it. So, and that person on the other side might have their phone out. The camera's not facing them. The person on the other side might have their phone out and be looking stuff up and Googling it and ChatGPTing things and have the notes open and the textbook and everything. So I don't have any respect for it. And I feel like people who are trying to learn and learn how to learn and actually apply that stuff and show me that they learned how to learn, it doesn't show respect to you guys either. So I do my test taking in person. It'll be in the classroom. We do not have labs on my lecture exam days. Okay? I can't do the same for the lab exams, and I apologize for that. I tried to work the schedule around it. I just can't fit everything in. But on lecture exam days, we won't have labs. So we have 4 lecture exams. We have 2 lab exams. So at least those 4 lecture exams, you don't have to worry about a lab that day. Okay. It also means that you can take your time on the test, because we always have a cushion of the lab after that, right? And I don't time my tests, and you won't need me to. It's only 50 questions. 35 of them are multiple choice, 15 of them are short answer. It's an essay question, but like really you can answer some of them with one word, and you shouldn't need more than 2 sentences to answer those questions. I grade them by hand, and I do have partial credit on those. So the short answer ones are worth 2 points, multiple choice is worth 1. Short answer, you can get partial credit, and I don't just mean partial credit. Like if the question is, what does peptidoglycan do in a bacterial cell? Well, we actually already mentioned that earlier. It's part of their cell wall. But if you put, it has something to do with the outside of the bacteria structure, but I can't remember what. If you type that in there, you'll probably get half credit, if not 75%, because hey, you get the idea of what this is for. So you might not get 100% credit, but you could get 50 to 75. But if you don't remember anything about peptidoglycan, and you're like, but I remember she said that when we test starch, we add iodine to it in the lab. So you could type, well, I don't remember peptidoglycan, but I remember we added iodine to test for the presence of starch in the lab. Then I'll give you half a point for that. So you get 25% credit just for saying another fact. Can't reuse the facts on any other tests either. So you can't reuse them from test to test. And it can't be something that I already tested on. That's my only rule. Or you can get some half points there. And that is something. It's worth more than you think. Okay, so we have that leeway with that's why I actually like the short answer questions, because it gives you guys an opportunity. I might not know this fact, but I learned some things. Let me share what I did learn with you that you didn't test me on. So opportunity there, which has helped quite a bit with my overall grades with that, especially after the loss of the cheat sheet people. Okay, so we'll say that. So yeah, so that's what you can expect with the tests. For the layout of the course, again, here's your first up stuff. Knock out everything here, and you'll be able to open up note-taking skills. It just has some info on how to take good notes and how to study properly. But then it has a little quiz after that. If you complete the quiz, you don't even have to get all the questions right, okay? If you complete it, I'll give you a 10 out of 10. And then that replaces a chapter quiz. So that's one chapter quiz that you won't have to worry about, okay? All right, so if I come back out of this, leave student view, the rest of this stuff is going to be set up in the weekly modules. And they're set up with an overview section, an overview of lecture stuff, and lab stuff. Now, week 1, I have all of the stuff there that you could possibly need. Like, it's all the resources you could need. But if we look down at week 2, all I'll have is the learning objectives. And every week I remind you about the disease case study project. This is your project for the lecture side. For the lab, I have the lab manual and the lab learning objectives. So this is your lab manual. That a lot of people are asking me about, like, well, how do we access the lab manual? What is the lab manual? Where do I buy it? And whatever. No, there's a PDF document of the whole lab manual. And in lab, we have a dry erase version of that exact same manual. So you can print it out if you want to. If you do print it, I recommend doing it in color. Even paying for the color in the library to print this out would probably be less than what you would spend on an actual lab manual, honestly, if you want a printed out version. But if you're just going to use the digital version, which I recommend, you can use the digital version. Or you can just use the dry erase ones in lab. And dry erase in lab will work through the lab. You'll answer questions with it, and you take a picture with your phone, upload the picture of your answers, and I grade that. So you don't really need to print out the lab manual unless you're just that person that likes to have a tangible one. You're welcome to do it. But the whole lab manual is there. Here's the lab learning objectives. Now you'll notice I have lecture learning objectives. I have them set aside for each unit. And then if you were to click on it, it has for each chapter what the learning objectives are. For the lab, I have learning objectives. Anybody take a class with Dr. Shearer yet? Okay, some people are in it now. Okay, so Dr. Shearer has learning objectives, but he doesn't test from them. Just warning you guys, for those of you who are in it. And if you've taken his class, then you know. Okay, he doesn't test from his learning objectives, but I do. So all of my test questions are derived from the content from my learning objectives. So I click on my learning objectives, all of them. I quite literally sat there with my learning objectives up, and I look at the learning objectives, and then I write a question based on it. Literally, all of my test questions are like that. So you can go through the learning objectives. Don't just memorize the answers, right? You can probably do okay. You can probably get a C if you just memorize the answers, but you need to be able to relate them and actually work with them, right? So it says compare and contrast eukaryotes, and acellular microbes. We'll actually be doing an activity with that today. So that I don't want you just to be able to define each of them and just have that memorized, but actually be able to have a discussion about that. So if I ask you in an open-ended question, compare and contrast these two, then I expect it to be, appropriate and kind of conversation about it, right? So in class, I'm going to be calling you out with questions kind of like this one. And I expect you to verbalize it back at me. And it's been shown, science has shown, if you can actually verbalize it back at me, then you had to think through the logic of the idea. And that can actually open holes on where your understanding is in those principles. So that's why we're doing what we're doing in the class. So we can start thinking through those things so that on the test, when you read questions like that, you're not stuck in that stress moment. You've already talked through this concept. You already know how to have this conversation and stuff like that. So there's your learning objectives. These are where I write my test questions. So it might say compare and contrast eukaryotes, eukaryotes, and acellular microbes. This would require you to understand about a nucleus and the fact that a nucleus is a membrane-bound structure. And if you have a membrane-bound nucleus, you are a eukaryote. Eu means true, karyo means nucleus. Akaryotes, A is without, like asymptomatic, without symptoms. So akaryote without a true nucleus. They still have a genome, it's just not in a nucleus. Not contained, it's just out in itself. And no, there's no membrane-bound structures in an eukaryote. And then acellular microbes. These are microbes that don't have cells. A cell is a whole membrane boundary. The whole thing has a membrane around it, and it's able to multiply itself without needing help from other structures outside of it. Viruses, for example, cannot do it. Viruses are just genomes with a protective layer around them. They're just a genome. The genome has nothing to help turn that genome into products contained in them, okay? So viruses get into cells and use all of the machinery from the cells to replicate themselves. But can they replicate themselves without that? No. And so the argument of whether or not viruses are even alive, that's a real scientific argument. We don't even know how we say that they're even alive. They can't replicate themselves without the cellular machinery. But cells are units that we know are alive. They can replicate themselves. If you cut your arm, your cells will replicate themselves and repair you, right? And so that's how we define those sorts of things. But if I were to ask you on the test, if I were to say something like a patient comes in with an illness, and whenever we culture the pathogen that caused it, the germ that caused the illness, it had cells with nucleus in it. Is it eukaryote, or acellular? Now, memorizing that stuff, you have no context on how to apply that, isn't going to do you any good in that kind of question. But that's the type of question I might ask. You get what I'm saying with that? To understand your learning objectives. Don't just answer them, right? All right. This type of testing, the way that I have my learning objectives and I test from understanding the learning objectives, you are going to have that in the nursing program. So they test from their learning objectives. They write NCLEX style questions based on their learning objectives, and that's what they're going to test over. So be prepared for that. And that's why I do my class this way, because this is what you're going to see in the nursing program, and that's where I want you to have the real success. I'm preparing you for that. Okay. And those, like I said, those of you going into vet tech, this line of learning will still help you. Okay. It's still going to help you. It's still going to be very, very useful. So this is the idea. So there's my learning objectives. You've seen them. Cool. They're there. You can access them anytime you want, and they'll be in all of your reminders about what you're supposed to have learned that week and everything. This is the slowest. So we'll have the disease case study project. This is a presentation project. You'll also have an assignment. There's an assignment that's not for a grade or anything, but it just is an assignment that's there because I know a lot of people just follow the assignment. Like, I'm not stupid. So if you're an assignment follower, there's a whole other assignment just to sign up for your disease. And it'll remind you, sign up for your disease. This is the day that you have to have signed up for it. And that's a whole assignment in and of itself, a completed or incompleted assignment. And then the assignment that it goes with is this disease case study. So you have about a month to figure out what disease you want, just so you know. There is a deadline for that, and it is on that other assignment. But you'll click the link to sign up for the disease. And look how we already have some people signed up for diseases. We have somebody signed up for African horse sickness, somebody for AIDS. If we were to scroll down, there's pretty like we have canine distemper has been chosen and stuff like that. So there's so many diseases on here. You can already see this is alphabetical. And so you can scroll through and find a disease that you're interested in. But since AIDS has been chosen in another of my sections, you guys can't choose it. So it's like all of my sections sign up in the one place. Okay. And so you guys can find a disease that you want to present on. The actual assignment for this is outlined here. You will put together a presentation where you will find a case study or a review of case studies, and you will present it at the beginning of your presentation. Define any terms related to it, and then the rest of your presentation is talking about your pathogen. So what is the organism that causes? We can talk about malaria, for example, right? So malaria, we know is a disease most of us have heard of. It's transmitted by mosquitoes, right? But the pathogen that causes malaria is Plasmodium. It's a different name. So knowing the name of the pathogen that causes the disease, tell me about the pathogen, what type is it, what's its virulence factors, those are things that help it cause infection. Whether it's modal or not, that's a virulence factor. Whether it has a capsule to protect it from being eaten up by white blood cells. And these are all things we'll learn about throughout the semester. Then signs and symptoms associated with that. To be clear, a sign and a symptom are different things. We often say, oh, you have a cold, what are your symptoms? But a symptom and a sign are different. Signs are things you can measure, and symptoms are things that are just perceived or reported. So when you're having pain, when you're in a lot of pain, can I measure that in any way? No, but you can report it to me. That's a symptom. But a sign, and I think of it as like a stop sign, is something that's tangible that you could hold, right? A sign is something that you can measure. So a fever, can you measure that? Yeah. Nausea, though, can you measure nausea? No, but can you see vomiting? Yes. So nausea is a symptom, vomiting is a sign, because you can verify it. So that's really, they're different things. Risk factors associated with it, people who have immune system issues, are pregnant, little babies, and old people, they all have lowered immune function. So they're often more susceptible to diseases. So that's a risk factor. If you're an IV drug user, that's a risk factor for some of the diseases, right? And so depending on what disease you choose, those are risk factors. Modes of transmission, how is this thing transmitted? Through IV drug use, through blood exposure, through fecal oral transmission, through air droplets, whatever it might be, mosquito bites, whatever it might be. How is it diagnosed? So this is something I want to make clear, and I'll probably re-go over it again when we get closer to the due date. This isn't due till November, okay? But diagnosis, I don't want anybody reporting they do a throat swab. So this is why. Y'all telling me about how if you have a sore throat, somebody will be presenting over the flu. Like, oh, well, they do a swab, the nasal swab. Cool. So I can swab my nasal cavity too and then throw it in the trash. That was a nasal swab. That diagnosed my flu. What do they do with the nasal swab? Y'all. So we learn about that stuff in this course. We haven't yet, but we will by the time that is due, right? And so I expect you guys to tell me, what tests do they do on the nasal swab? They do a nasal swab and then they perform PCR to look for the genetic information for flu. That is telling me about the diagnostic methods. That makes sense? So I want you to tell me about the specific diagnostic methods and how they're used to diagnose that thing. Common ones. If it's a rapid test, don't just tell me they do a rapid test. Does that rapid test for antigen or antibody? Like a COVID home test. COVID home tests are antigen tests. What does it mean to test for an antigen? I don't expect you to answer that question yet. But I do expect you to answer that question by the end of Unit 3. Okay, so that's the type of stuff that we'll be talking about. That'll be in your presentation. And then what patient care, treatments, or vaccines. Some diseases like a cold, but if you get a cold, like what's your treatment? What we would consider like home care, comfort and things like that. Like you might take some Tylenol, but you'd still put that in your presentation, okay? If there's a vaccine, if you choose the measles, then I expect you to talk about the MMR vaccine, right? So whatever's appropriate for your disease. There is an example of somebody that did an excellent job over the summer. So this is a recent example. It is a recording in PowerPoint. So if you download this file and then open it in PowerPoint and then play it as a presentation, it'll have her voiceover with everything. It's 7 to 10 minutes long. I'm not asking for a whole hour-long presentation, okay? 7 to 10 minutes. You might have more information on your slides than what you review on your presentation. But as long as you've got all your boxes checked on the info thing, your presentation doesn't have to be reading the slides. In fact, you actually get graded poorly if you just read your slides. Don't just read your slides. Don't rely on notes or scripts either. Because you do get graded on it being conversational. Because if you did your research on your disease, you should just be able to tell me about your disease without referring to it in notes. Now, referring and reading some from slides or notes is okay. But there should be some aspect of it being conversational. Show me that you actually learned the thing and aren't just reading a script, right? So you do get grades on that. It does have to have citations. Citations have to be in APA format because that's what the nursing school requires. Okay. and you learned it already, and I hate it too, but that's just how it's going to be. There's a rubric for grading this stuff. If you're covering all your points, or if you're just covering part of them, and how good your presentation is, each section has a score associated with it. And that rubric, as big as it is, but all the information is there on how I will be grading you on that, so that when you get your scores, you'll know why you got the grade that you did, and et cetera. If you get these, like you get your PowerPoints put together early enough, send them to me early enough, and there's a deadline, there's an announcement for it and everything. But it's basically like 48 hours before the due date. If you can get it to me before that 48-hour deadline, then I will look over it and tell you what's wrong with it. And there's no reason why you can't get full credit on it, right? If you get it to me early enough and you get it done early enough, I'll email you back and be like, you're not talking enough about the diagnostic methods, and you're not covering this enough, or tell me more about the virulence factors. You go back, you make edits, you resubmit, and now you should be set up for full credit on it. So that should be easy points, okay? Now, you do have to present in person. So there's part of that too. So you either present in person, or you can record it like the student did on this. You can either record it in PowerPoint, which I have directions on how to do that, or you can record it as like an MP4 if you use like Canva or something like that. Record it as an MP4, but it has to be submitted in either MP4 format or PowerPoint format. Those are only two formats that I accept. If you're doing just the slides and you want to present in person, which I recommend, I'm just a person, I don't want to watch a recording of me speaking, because if you do pre-record it, I play it in front of the class. Okay, so just be aware. So whatever you want, it's on you to make the choice if you want to do it in person or record it. But either way, it's going to be in front of the class, and I'm going to grade it. If you choose to record it or present it in class, I reserve the right to ask questions about your presentation during it or after it. Okay. I may or may not. If I'm getting a sense that your presentation is very scripted, then I'm more likely to ask you questions. But if your presentation was excellent and you are talking about it like I'm talking about with you right now, without referencing a thing, and you're telling me about the disease and without much reference to the notes, I'm less likely to ask you a question. So, okay. But that comes in November. And then all of Unit 4, instead of me calling on you as individuals with questions from the Kahoots and the activities, in Unit 4, it's just going to be you guys presenting. There won't be any of the other activities. We have the Kahoots that you guys can still practice with on your own time, but we probably won't be doing them because we want everybody to have time for the presentation. That makes sense? Okay. So that's that. Back to the modules. Let it load. You gotta be patient with this. All right, other stuff. We have the preview section. So we've gone over the overview stuff, the big picture stuff on there. We have a lab project too. We'll go over that in lab. All right, we'll do the lab stuff in lab. Preview stuff for lecture. We have extra credit. This is those chapter connect homeworks. You know the ones. Almost all of us have had a course that has McGraw-Hill Connect on it, and they have these chapter homeworks. I generally don't have a ton of respect for them, but I do have mine set at the lowest setting. So it's like, should be anywhere from 15 to 30 minutes for most of them. None of them go over an hour. There is a couple where the concepts were heavy in that chapter, so I have a little bit higher setting on them. Anyways, those are extra credit in my course. You do not have to do them, all right? If you want to and you find them useful, then please feel free to and get some extra credit. You can do as many or as few as you want. They're extra credit, right? For the learn part for the course, this is the concept map. This is also where you'll find my lectures, my recorded lectures. So if you click on these, that's my learning objectives, what you're supposed to be learning, then use my lecture and my slides. You see there's slides there too. Right? So my slides that go along with the lecture, use those to figure out your learning objectives. So I don't lecture outside of the learning objectives unless I feel like it's helping feed into understanding of the learning objectives. I test from the learning objectives, but the lecture is helped to explain the content in the learning objectives, okay? I never test from the textbook that isn't in the lecture and the learning objectives. People often ask that, it's like, how much do I need to read the textbook? Use the textbook to supplement your understanding. If the lecture isn't explaining the learning objective to you, go look the learning objective up in the textbook and see if it helps. If you want more clarification on a topic, use the textbook for that. If there's information in the textbook that isn't in my lecture or my learning objectives, you will not be tested over it. I guarantee it. Okay, so you can rest on that. You can hold me to it. Okay, so there's my lectures. They are pre-recorded from previous semesters when I lectured in person. All right, If you have any issues understanding something or hearing, because I have a tendency to walk away from cameras, if you have any issues understanding or hearing, please let me know sooner rather than later. Or if you can't figure it out on your own, what I was talking about, please let me know. And I'll see if I can find a better recording or if I can just like explain what was missing on that. And you let me know. And that way I know there's an empty spot in that lecture and I can at least add a note on there on that lecture for everybody. You know what I mean? So I've edited them down, but like your perception of what you can hear and not, I don't know what your computer setup is. So just let me know. And then it explains the assignment for the concept maps. They're a concept map. Like, I don't know what else to tell you, right? So it's a map of how the ideas are related and then a brief explanation of each of those ideas. And so this is one that is complete for chapter 1. And you can copy this one for chapter 1, but you'll have to make your own from here on out. I don't have examples for each chapter. I only have one for chapter 1. But this is what I'm expecting with the concept maps. So you'll have to do chapters 1 and 3 on your own time, okay? But I will say this too. I dropped the 2 lowest concept maps. So if you don't want to do chapter 1 and 3, you don't have to. Just make your notes or something. It'll drop the 2 lowest concept map notes, things, scores, okay? So there's the concept maps. This is what I expect for, this is a student that ended up getting an easy A in my course, and all that. Even since I changed, because I changed all my tests, right? And I have a class over the summer. They had a new test, brand new tests, right? Brand new test, no cheat sheet. They had to do concept maps. Nobody else's concept maps that they had to look at. They had to make their own. They had all of the other assignments that you guys are going to have. And in my summer class, it's a hybrid class, which I think is harder because you don't get interaction with me about the lecture material. It was hybrid class. My hybrid classes during 16-week courses, by the way, usually do it a letter grade lower than my in-person classes. All the same assignments, exactly the same due dates. Everything's exactly the same. I even post everything that I do, that I record for them. So there's no excuse for them to do lower, right? Except we know why they are doing lower, because it's their responsibility to stay on top of the things, right? So anyways, they usually do a letter grade lower. Well, my summer class this summer, they all got A's and B's. If they can get no C at all, not even one C, no D, no F, every single person got an A or a B. There's no excuse for you guys. You get 16 weeks to do what they did in 8. They met in lab every single day. Every Monday, Tuesday, Wednesday, Thursday, they met in lab. And you guys don't have to meet in lab twice a week. You only have to meet for lecture once a week. If they can get all A's and B's, you can too. You can. They're not any smarter than you, right? So you know what I'm sure you've heard. This class is a hard class. You just talk about it. And I usually, in my 16-week courses, expect most of the class to get C's. Why? Why is my summer class getting A's and B's? And I think we know why. But it's the same for them. They get the same course. They just have to do it in 8 weeks. You guys can do it. Just keep on top of the work, right? Keep on top of the work. Keep on top of the work. I'm trying to help you guys out where I can with that, with all of my reminders. But really, so anyways, anyway, so that's the concept map. And then in class, we will be spending class time with the application part. Oh, SU, okay, see. In structure. You add this to my favorites. Okay, okay, back to it. All right, so back to it. That's the stuff that you'll have to do before class. So you'll actually have a due date for chapters 2 and 4. It will be due by Sunday at midnight. So you'll have your concept maps made for chapters 2 and 4 by Sunday. So on Tuesday, when we come in to do this again on Tuesday, we'll be ready for the conversation. We'll be ready for me to call you out. And you'll have seen the material and you'll have organized it. And you can use your notes when I call on you. It doesn't have to be out of recall. Use your notes, look it up, take the time, talk with your fellow students. It's not a test. It's meant to be conversational and discussional, okay? So that's what we're doing in we did preview, we did learn, this is your concept maps. Apply for lecture, this is our lecture application activities. We'll be doing one today. This is chapter 1. There's some questions here. Which of these scenarios has a eukaryote, an akaryote, an acellular, or no pathogen disease? And for chapter 3, we'll be going over a case study. and learning about chapter 3 stuff and how it applies to an actual case. Okay, some of these guys will have like chapter 4. When we do these activities in class, it's supposed to open automatically, but it's not. This is an actual scientific paper case study. Okay, so that's this, most of them are going to be like this. For the beginning, I like to ease you into that and not just jump straight in, okay? So most of them will have an actual, and it's usually just one page from a whole case study, but it usually has all the information we need to answer what I'm asking about it, okay? And so that's what we'll be doing in class. In addition to, and these are available on Canvas, we'll always start out each day, and we're about to get started with it now already, because we have an hour left, but 50 minutes really. In those lecture application activities, it's 2 parts. There is a Kahoot quiz, which we'll do in class in person. And that's where I'm really going to be calling on you on the Kahoot quiz. We'll have a question. It'll give it, everybody will answer it on their Kahoot. And then it'll give us the answer. And then I will call on you to either explain why that right answer is right or why one of the wrong answers is wrong. And we'll knock out everybody in the Kahoot quizzes because those are easier questions. But those other exercises with the worksheets, those will be working through conversationally, and they won't be as much like calling on you with those, okay? But if, let's say I call on you on a Kahoot quiz question and you don't know the answer to it, if you know the answer on the worksheet part, chime in and get your points then, right? So you make up what you didn't know on a Kahoot quiz if you know it on that stuff. Does that make sense? So we'll do that today so you'll get a taste of it. This is the in-class activities. I will put the grades in for these. You don't have to submit anything for these. I do all the work on that end, okay? Online class is different. They have to do all of that on their own. All right, so that is the apply. And then we have the assess. We have quizzes for each chapter too. So every chapter has optional extra credit, connect homework, optional. The concept maps, not optional. The activities that we do in class, not optional. Quizzes afterwards. There are only 10 questions, multiple choice. It's timed 15 minutes, but it's not on lockdown browser. So you can open up your notes and search through them, but I time it so that you have to have some reference on how to look through your notes. You have to have exposure to the material and kind of have a concept of how it's organized in order to take the quiz. So there's 10 questions, and honestly, nobody ever has any problems with the quizzes. Then we have, but if you do your chapter, your note thing, it'll replace one of your chapter quizzes. Then we have the exams will appear in the assess also when we have an exam that week. Okay. Then we have review materials. These are all not required. They're study guides and things like that if you like that sort of stuff. So that's all in the modules. Every module looks exactly like this. If there's nothing for something, like if I don't have a chapter for extra credit for that week, it'll say NA. So there'll always be something there to let you know if you're missing something or not missing something in the modules too. So if you're a module person, all that stuff is there. On the assignments, one last thing before we get going. So I told you about my exams, right? 50 questions, 35 multiple choice, 15 short answer. Short answer is worth 2 points. Multiple choice worth 1. I don't time my exams. Don't let the time be the thing. Also, during my exams, I usually play soft music that doesn't have lyrics. So we're talking like classical music or lo-fi, like soft, because I have ADHD and many of you do too. And so for me, having that background music actually helps me not be stressed out by the silence. I get super stressed by the silence. But if you are somebody that doesn't want the music, just email me and we won't have it. And everybody in here that wanted the music, they'll respect that. Okay? But generally speaking, I find that people want the music. So I still tend to start with that. And if you are a person that doesn't want it, just email me. Nobody has to know it was you. Okay? You just won't have it and you just won't have it. And that's that. Okay. So low level music. I will dim the lights. Usually this is the level of lighting to expect in this classroom. I like it a little dark. And I will usually put up like some jellyfish or something so that you can, if you want to look up from the test and take a deep breath and recenter yourself. Don't let the anxiety take over. And I usually also have rosemary oil that I diffuse. Anybody allergic to rosemary that they know of? Okay. Because rosemary has been shown to help with memory recall scientifically. So I do rosemary oil and I diffuse that during. I do what I can for y'all. Okay. Yeah.
specific to Rose.
We won't do it then. Yeah, we won't do it. That's not a problem. No, not a problem at all. We can respect that. Yeah. So I'm glad that you tell me that. That's exactly why I want you guys to tell me now, so that I don't put you in a more stressful situation, right?
Yeah.
Exactly. Yeah. So we won't do that. We won't do that. Not a problem. I feel like even just having the jellyfish up is better than what most professors do. So, but it will be on lockdown browser, and I'll be walking around kind of just checking on everybody now and then. And I have a rule too, other rules that we'll follow during that course. I actually have to have you have your phones out during my test. Okay. So your phone has to be out where I can see it. It'll be on the other side of your laptop or tablet, face down the whole time. I need to see your phone. And if you have a smartwatch, it comes off and it sits next to your phone. I need to see where it is, because I have seen students doing this move during the test. Okay. We're not doing it. So anyways, so we have a clear understanding of the lecture part. We get on that. Okay. I know you're still kind of wondering how this thing is going to work with the calling on people. So we're going to do it now. This is, yes, start. So Kahoot.it is where you'll go on your website, or if you have a phone or the app on your phone, you'll enter in the PIN when you get to Kahoot.it or when you open the app, or you can use the QR code. This is how I'm going to check your attendance as well as your participation. So you do have to log in, and please use your email when asked to sign in. Please use your email, some email that has your name in it. It doesn't have to be your OK State email, but something that has your name or some part of your name, so I can identify who it is. I have a little sound in the background.
I'm signing up right now if I haven't done it
yet. Yes, probably. We'll wait. I think I'm going to count whoever's in here, make sure everybody gets signed in. 2, 5, 6, 8, 9, 10, 11, 12, 13, 21. And I am going to call on you today. Maybe not everybody, but we're going to have an example of how it's going to work. Okay. I think I counted 21. There's supposed to be 24 people in the class. We already know we're going to have 3 absences. Yes, 21. So yeah, so once you get signed in, we'll get going with this. We'll do the chapter 1 Kahoot and the chapter 1 activity, and we might just jump to the chapter 3 activity, depending on what time we have left. Okay. Kahoot links are always available on your lecture activity exercises, so you can always go back to them and practice the Kahoot links. Just use a different, for you guys, either a different email or a different username. It just depends on how that link works. I think just username will be different, whatever you're putting here. But you're welcome to replay them as many times as you like. I do weekly reviews also. Every week on Thursday at 4:45 p.m. I hold a live Zoom where I will review the content for the week for the lecture part of the course. And I also go over the most missed concepts, usually 2, maybe 3 for each chapter that was most missed on previous exams. And you can bring any questions you have to those reviews. If you can't make it to those reviews, I do record them and post them in the discussion board. So it's okay if you don't make it. I'll still have them if nobody shows up. So don't worry about it. I'll just post previous recording if that happens. All right. Everybody make sure you're signed in. I've got 17. We have 21 students, so I want to make sure everybody's getting signed in. Make sure your name is up here. This is where I'm going to count your credit for participating in the class today. Otherwise you get a 0. That'll be already a grade we don't want to get. So it doesn't matter what your score is either. If you've completed this in class with me, then you get full points for the Kahoot part of it. Okay. It's the answering the questions when I call on you. That part might vary depending on your research. So we've got 2 people. Everybody ready to go? Yeah. So if I ever start, because I can't always like wait for everybody. So normally this is how our day will start. We'll start with the Kahoot. I'll give maybe a reminder about a test coming up or whatever. Then we'll get going with the Kahoot. That'll be usually how our day will start. Okay, first chapter that we're doing is chapter 1. So we want to make sure that we get this process down smooth so it doesn't take too long. But if I get started and you haven't started, you're late, or you didn't get joined in before we started, you can always enter it in that code. It'll always be like at the bottom of the screen or something, and you can join it. You can also do class via Zoom if you want to. As long as you're joined in and you can participate and you can answer when I call on you. I don't care if you're on Zoom or here in person. If you have some reason to be absent for class, let me know and we'll work something out for you. But I do require like excuses, like verifiable excuses. So a note from the school saying your kid had a fever or something like that would work. Or if you go to the doctor, a doctor's note, or if you have a court case, something saying that you had a court case that day, just whatever. But I do have to have some sort of verifiable evidence for that stuff. Okay, are we ready? Because we're gonna get going. Even if we're still signing in, you can always join in the middle of it. All right, chapter 1. First question. Do your best to answer these, okay? And I'll go over them. How do we define a microorganism? And I will call on somebody. Okay, so our answer is it's something that's too small to be seen with the naked eye. So my first person I'm going to call on is Brandon. Who's Brandon? Okay, so you're Brandon. All right, Brandon, why isn't it bacteria? Why is bacteria not the right answer?
Might be some bacteria that you can see. I think.
So we're getting kind of in that thinking, but we want to think a bigger picture, right? So he's thinking about it, when he's thinking through it. So I probably give him credit because he's trying, right? He's talking through it. The reason it's not bacteria, when we see anything too small to be seen with the naked eye, right? That includes bacteria. Bacteria we can't see with the naked eye, none of them, right? But there's other things that we can also not see with the naked eye that might be considered a microorganism. So that might be mold spores, that might be viruses, that might be protozoa or amoebas, like what causes malaria is plasmodium. There's other things that can be not seen with the naked eye that aren't bacteria, right? So this is too narrow. This is a bigger group. That makes sense? You guys see what I'm looking for when I ask you this question. All right, so we're going to move on to the next question. I'm going to call a different person. Viruses are made of eukaryotic cells. Is this true or false? I may have mentioned this. Viruses made eukaryotic. You can see the little pin number down at the bottom. So we're split 50-50 here. So the answer is false. So it is false. So hopefully whoever I pick on here has some idea why that is. So Tawny Collins. Okay.
Viruses are acellular.
Yes, they are acellular, so they can't be made up of any kind of cell. So I mentioned that earlier. But again, when you guys have done your concept maps, so for today we're just kind of guessing and stuff. When you guys have done your concept maps, hopefully you'll be able to answer these questions with a little bit more background. That's okay that we're just guessing today, right? So we've got an idea of why this is false. Viruses aren't made of cells at all. So they aren't made of eukaryotic cells even. Eukaryotic cells being the ones that have a true nucleus. Chapter 1 will also tell you that bacteria, they don't have a true nucleus. So they are akaryotic. We have akaryotes, eukaryotes, acellular pathogens, which are like viruses, and prions, which are misfolded proteins. All right, next question. Question 3, which came first on Earth? Akaryotes, no nucleus, or eukaryotes, they have a nucleus. Chapter 1 does cover this. But let's make a guess here. Which do you think came first? Right. Yes, the chicken or the egg, right? So it tends to be the akaryotes. I'm not going to call anybody else probably for the rest of the day, but you guys get an idea of what that's going to be like now, right? So we'll just go over some of these questions for now. But akaryotes, because we started out and fossils and scientific evidence shows that the cells that exist first, ones that did not have a nucleus. There's actually evidence to support that. What happened was something like a bacteria, no nucleus, ate up another thing like a bacteria that didn't have a nucleus. And then that became a beneficial relationship for those two cells to have one inside of the other, and that became a permanent relationship. Now, whether or not you see that developing as a nucleus or not, we can argue or not. But mitochondria, So you guys know what mitochondria are, right? The powerhouse of the cell. Most people at least know that. It's actually where cellular respiration. So mitochondria have their own genome. It's separate from your genes. Your genes don't code for your mitochondria and nothing for your mitochondria. Wild, right? They have their own gene. Our genes, we know as humans and as animals, any animals, they have linear genes. We have those X's and Y's and things. They're linear. And we have pairs of them. Bacteria have one chromosome and it's in a big circle. Guess what your mitochondria has? One chromosome, big circle. It also has its own ribosomes that make proteins for it. And its ribosomes look like bacterial ribosomes, not like the rest of your cells' ribosomes. Wild. It also has two membranes. It has a membrane on the outside and a membrane on the inside, which is something that we see with some of our bacteria, not something we see in our actual overall cells. So there are things that relate mitochondria. They even look like bacteria, y'all. So there's things we can say mitochondria probably used to be something like a bacteria back in the day that another cell ate up. That little bacteria made energy that that cell could use. And that little bacteria got a house and protection and food for it. So that became a symbiotic relationship. So that theory is called an endosymbiotic theory. And there's a ton of evidence supporting it. So your mitochondria have their own genes. Did you also know that all of your mitochondria in your cells, whether you're a male or female, every single person on the planet, your mitochondria came from your mom's side. The sperm is too small to carry mitochondria with it. So the egg carries mitochondria with it. And that mitochondria has its own genome. So it replicates itself. So everybody's mitochondrial DNA comes from the mom. We call it maternal mitochondrial. It's true. Cool stuff, right? All right. So we're gonna I'm gonna stop here with this quiz. And we're gonna jump to, normally we'll go through the whole quiz and I'll call on people with all the questions, but I want to show you guys what it would look like if we were doing the assignments next. We do the quiz and then we would jump to this assignment. So the actual assignment associated with this is our activity stuff. This is where I'm less likely to call on you, but I still expect you guys to participate in the discussion, okay? So we are gonna categorize whatever is causing the illness. We're gonna categorize it as either eukaryote, akaryote, or acellular. And I'm going to put some notes at the side so we have a reference, okay? Eukaryote has a true nucleus, meaning that it has a membrane around it. Akaryote, no true nucleus, no organelles. We'll talk about what organelles are in later chapters, but those are things like mitochondria, endoplasmic reticulum, whatever. So bacteria don't have any of those, all right? And then acellular, literally they have no cell aspects to them, not cells at all. And then we have diseases that are not caused by pathogens. And I'll just put example like diabetes. It's not caused by a pathogen, but it is a deviation in health, so it's still a disease. All right, so we got something to refer to now. First question, a patient presenting with sore throat had their throat swabbed and cultured. Lab results indicate the pathogen is small, round cells arranged in clusters that appear to lack a nucleus or organelles. Would this be eukaryotic pathogen, akaryotic pathogen, acellular, or no pathogen? Right, akaryote, right. So this will be probably bacteria, probably. All right, a patient with severe headache, stiff neck, fever, and photophobia. Photophobia isn't fear of the light like it implies. It's light sensitivity, okay? They've got light sensitivity, they've got a stiff neck, a fever, and a severe headache. What is this already? What disease would we call that? This is meningitis. It's meningitis, right? So that stiff neck is what gives it away with that. And so already this is meningitis. So you guys are aware, right, that meningitis can be caused by a lot of things, right? So meningitis is defined by the signs and symptoms. This is something that we call a syndrome, not defined by what causes it, like a flu. The flu is caused by the flu virus, but meningitis can be caused by a lot of things, but the signs and symptoms are generally lumped in one big old category. So this person has meningitis. We do a spinal tap. We look at their cerebrospinal fluid, their CSF. And it looks like the lab shows that in the CSF, which should have no cells in it at all, except maybe your own cells. We've got a pathogen, and that pathogen has branching nucleated cells. Large nucleated branching cells is the pathogen. So is this a eukaryotic pathogen, akaryotic, acellular, or no pathogen? Eukaryotic, right? So the pathogen, I'm telling you, it has nucleated cells, so it's a eukaryote. And if they're branching like this, I can actually already tell you what type of pathogen it would be. It would be a fungus. It would be a fungus. So fungal meningitis here. Most commonly caused by not Coccidius. I always want to jump to that. That is a cause. Cryptococcus neoformans. Cryptococcus neoformans. We don't see this a lot in healthy people, but we see it quite a lot in immunocompromised people, homeless people, people living in communities where health overall might be poor. And it's transmitted in bird droppings. So environmental exposure. And we see this quite a lot in people with HIV/AIDS. To put AIDS into perspective for you guys, 50% of adults in South Africa have HIV. Half of all the adults in South Africa have HIV. Please don't close your mind to the fact that you live in the United States. When I start talking about helminth infections, these are parasitic infections, the parasitic worms, right? Despite what TikTok will have you believe, it's actually incredibly rare to have a parasitic infection in the United States. Really it is. The doctors aren't lying to you about this. This is fact. It's actually really rare to have parasitic disease from the United States. The most common parasitic disease worldwide is Scaris lumbricoides. We don't have that in the United States. And we're talking, this is a very common illness in underdeveloped countries. But why don't we hear about it or know more about it or hear about ways to prevent it or anything like that? Because it doesn't affect Americans. Americans don't care. Like quite literally, there's no funding to study these diseases. So that keeps it in perspective. It's not just about the US, it's not just about your mama, and it's not just about the upper middle class or the upper class, right? So you ain't gonna be treating, most of the people you're gonna see in ERs for sure are gonna be lower class. So they're gonna be the homeless people. Let me tell you, I worked at the lab at Integra Southwest when I worked with BLO. Most of your patients are gonna be hookers. Okay? I'm just saying, and they all got tricked. I'm just saying. So anyway, so next one, number 3, a patient presents with a runny nose, a mild sore throat, mild fatigue, low-grade fever, what we might consider something like a cold or flu-like disease, mild flu if it is the flu, but something like a cold. We do a nasal swab. The cultures that are performed in the lab show there's no visible cells that are the pathogenic cause of this. Might be like, normal biota would have grown up in the culture, but nothing indicating that the pathogen is made of cells. No cells, interesting. Despite indication that the patient's illness was contracted from another family member who got sick first. Right, so it's something that is contagious, but no cells were present. So what one would this one be? Acellular. So what do we think it probably would be? Probably a virus. Yeah, like the cold. A patient at your clinic diagnosed with type 2 diabetes, blood work supports that. There's no pathogen associated here. Everything just seems in line with that. That would be no pathogen. Exactly. And then our last one, James visited the UK in 1992. 20 years later, he showed a relatively rapid mental decline and a significant dementia, despite being only 30 years old. After his death, approximately 1 year after his symptoms began, autopsy indicates that his brain tissue is riddled with holes like a sponge, but cultures show no evidence of cellular pathogen. So this could be acellular or no pathogen, right? This one's a little trickier. So if we had gone over all of chapter 1, you would be more likely to choose acellular. And this is why. This is a disease that is not caused by a virus, but is caused by a prion. Prions are just misfolded proteins. Protein that does something normal in your brain, and then it sees a misfolded guy and says, I want to look like that. I don't want to do this anymore. I want to look like that. And it misfolds that way. If you ever read Kurt Vonnegut's Cat's Cradle, it's similar to Ice 9 in that. It's actually where the metal band gets its name from. But if you know, you know. But basically the idea is the protein folds one way to do its function. But when it sees the way the other protein is folded, it says, that's for me. That's the life. And it misfolds. And then other proteins that function normally see that, and they all just cascade and start misfolding. Now that they're misfolded, they can't do their function properly. And also, as I'm sure you guys learned in chemistry, in the body, what's most of our body made up of? Water. Water is polar, right? The H's have a little positive, the O's have a little negative, and things that are polar mix with polar things. But whenever you're folded, misfolded like these guys are, the hydrophobic R groups are now on the outside, and they don't mix well with water anymore. So they aggregate, and they cause holes to form in the brain. So when this is contagious like this, when we have a misfolded protein causing the other ones to misfold, that's a prion disease. It's an infectious protein particle, just misfolded proteins. And that's what this is. And so this is the case of something in particular. Does anybody know what this is called? Mad cow disease. Exactly. So when people get mad cow disease, we call that variant Creutzfeldt-Jakob disease, or VCJD. CJD can happen genetically. You could just have a predisposition for your proteins to misfold that way. Some people do. It's incredibly rare. Incredibly rare. It takes about 30 years for these diagnoses to pop up. You eat the meat that has the misfolded protein. 30 years later, you'll get dementia. It looks like Alzheimer's. It looks like dementia. For these people, the only indicator is that they're younger than they should be for having those symptoms. And then usually within a year or two of having the symptoms start, they die. There's nothing we can do. Okay? That's why mad cow disease was such a big thing for those of you old enough to remember it. That's why it was such a big thing in the 90s. They killed all of the livestock in the UK because of fears about mad cow disease, because you died, and it is spread from cows to people. So people will be like, well, why don't we just cook the meat? Like, if you cook the meat enough, it's fine. Nope, prions don't die from being cooked. They actually can survive incineration. Yeah, just wild. They're extremely difficult to control. You can actually contract these from surgical instruments that have been autoclaved, as well as from dental instruments that have been autoclaved. They're sterilized. We've removed the viruses and the bacteria. But if somebody had a prion disease, it touched usually their nervous tissue for some reason, and we autoclave it, that prion can still be there and be transmitted to another person. It's incredibly rare, very, very rare, but it can happen and it has been documented. Okay, so those are prion diseases. It's just a misfolded protein. There's no genome for these guys, so they're acellular. But is it able to be transmitted from person to person? Yes. So we still study it in microbiology. This is why I went into study biochemistry, because I was interested in that.
I wonder if it's more common than
Yeah.
dementia and Alzheimer's.
Yeah. So Alzheimer's actually operates on the exact same principle. So Alzheimer's works the same way. It's just we don't have a misfolded protein causing them to misfold. They just misfold because of age. So you have a predisposition as you get older. If you have genes associated with Alzheimer's, or if you have environmental stressors that put that on your gene expression, that as you get older, your brain proteins will misfold on their own. So it's not contagious. You're fine. Yeah.
Lewy body's dementia, is it the same?
It might be, yes. So it's basically based on like those Lewis bodies that are associated with any form of dementia. The sense tends to be where we have aggregation of proteins in the brain causing holes and then dysfunction of brain function. But this is when we talk about prions, that's an infectious version. And so those things can happen naturally in the body, but when they're spread from person to person this way, then it's prion disease. There's other prion diseases for you vet people, something that applies to you. You're not just going to be dealing with cats and dogs. I hope you're aware of that, right? So vet people, you should be aware of chronic wasting disease. So in deer, there is an epidemic right now in this country with chronic wasting disease, which is a prion disease transmitted amongst deer. And you're supposed to, if you're a hunter, have your deer that you hunt tested for CJD. It's actually required that you do that, but people just don't do it. We are having increased numbers in Oklahoma of this prion disease in deer. Right now, it's not spreading to people, but it is spreading to other mammals for the first time in history. So it used to be only in deer, now it's spreading to like elk and other animals. And so we're worried that it could change to a form that could spread to people. That is propensity for that. So test your deer, have the deer tested, tell your loved ones to have their deer tested. And if they are chronic wasting disease deer, there's still no reason why you can't eat the meat. But I'm just educating you so you're aware of the spread of the disease and they can document that and track that better. Okay. All right, we good on that? So that's that one. Yes. 1 and 2? Yeah. So number 1, the patient had a sore throat and they had pathogens that had cells that did not have a nucleus. So that is going to be eukaryotes, cells without a nucleus, eukaryotes. And number 2, they had a headache and they had meningitis. Their CSF have nucleated cells, that's gonna be eukaryotes. Yes, the E one, yes. You're all right, yes.
Do you want us to write this on a paper and then just?
You don't have to submit anything for this, just you guys being here and participating, I'm keeping track of that. So that's why I asked you guys to do the Kahoot. That's how I know you were here, and then you'll get credit for participation. Today, it's just participating. In the future, I'm looking for right answers, and I'll be tracking that, like what your answer is. Now, if I tell you that's not quite good enough for the credit, then you know, maybe stick your hand up for another question during this part. And if you can answer it, then I'll give you your credit for the first part. Make sense? Yeah. Because generally I'm calling on you during the Kahoot stuff. Those are a little bit easier. This is clearly like a little bit deeper thinking than some of them. So it seems straightforward now, but it gets more complicated as we go on through the course. Okay. We're gonna go to chapter 3. We got a little bit of time. I want to do a couple questions on the Kahoot. So we're going to log back into Kahoot. Oh, geez, this is going to stay here. Delete this. I guess there's a thing, right, that I can do. They're all drawings. Okay, cool. All right. Where's the Kahoot? There it is. Chapter 3, host live. We're going to do just a couple questions on this again. Start. We got a little bit of time. And again, most days we'll do all the Kahoot and I'll call on you during that time. Then we'll do the worksheets. It's only ever going to be 5 questions on those worksheets, just so you know. And like I said, they're posted in that assignment if you ever want to go back to them for study purposes. I do record these, and I will also always post these recordings in the discussion area on Canvas. You can join in via Zoom if you want. The only problem is with the in-person classes, we always have lab right afterwards. So it's like, you still have to come to lab anyway. All right, just double check that you're logged in. Give you guys just a minute. And we'll go to do a couple questions from this one. And I might call on a couple people. I'll try to pick questions that aren't like too heavy, okay? But remember, when you guys get back on Tuesday, I'm calling on everybody. But you'll be better prepared. All right, so we're gonna get going with our first question from chapter 3. You might not have any background on this, that's okay, just guess, okay? Which of the following is not included in our 5 eyes? So you really need a little bit of background for this one. Just make a guess. Five eyes are tools that we use to investigate pathogens. All right, so the answer to this one is infection. So I would call on you and ask you this. This is new though. This is new. We haven't actually learned this yet. It's on you guys to go back and learn that stuff, right? So the 5 eyes are, first we have inoculation. This is taking that throat swab and putting it on some media to get it to grow. That's inoculation. Then we take the media and put it in an incubator where it's got warmth and moisture and oxygen if it needs it, or no oxygen if it needs it to be no oxygen. For example, Clostridium organisms, things that cause botulism, tetanus, C. diff, and gas gangrene, those guys are strict anaerobes. Anaerobe means without oxygen. They'll die in the presence of oxygen. And so we can't culture them in a normal setting. We have to put them in a place with no oxygen, right? So that's where you get those like blood tubes, like when we do blood cultures, you guys might have seen those bottles. We'll have an aerobic one and an anaerobic one. And you have to get the air out if you're doing an anaerobic blood culture, because it'll kill the organs, quite literally. So anyways, we have our first step is inoculation. Then we go to incubation. After we've incubated it, we might want to, if you do a throat swab, are you just getting the bacteria that causes the strep throat, or are you picking up the patient's bacteria too, right? So you need to know which one is which. And that's isolation. We'll learn about isolation stuff. It'll go over it in this chapter, but we'll also cover it in lab. It's going to be our first lab, I'm pretty sure. So this like quad streak plates and things like this, separating out the cells so we can get individual colonies and create pure cultures. Once we have the pure culture of whatever we think the pathogen is, now we can do inspection. These are doing tests, like maybe do a PCR test, maybe see what kind of media it grows on. Does it ferment this sugar or that sugar? Does it ferment anything at all? Does it require oxygen to grow? Things like that. And that's the testing part. And using the results from the test is the identification part. Was it positive on the PCR or negative on the PCR? Because it was positive on the PCR that tested for strep, it was strep. Just doing the PCR is one thing, but taking all the evidence together and saying this is strep because of the results of the PCR, that's the identity. Okay, so those are the 5 I's. Inoculate, incubate, isolate, inspect, identify. So those are all 5 I's. Infection is the one that didn't belong. I won't call on anybody for that. We're just going to do a couple questions. Really, just one more. Differential media. What does differential media do? We have 2 types of media, selective and differential. Well, we have more than that. There's enriched also. There's complex media. But selective and differential are 2 that are on the same kind of concept, using it to diagnose. They're different. Differential versus selective. Differential media is media that tells you the difference. So anything can grow on differential media, but it'll tell you the difference between things. So we have, for example, some media called mannitol salt agar. It's got salt in it that inhibits the growth of some bacteria. That's selective. We're selecting for the growth of certain things. But if you can grow on it, you might turn that media pink or you might turn it yellow. And then we can tell the difference if you turn it pink or yellow. If you turned it yellow, you made acid because the color indicator is pH indicator. You made acid because you fermented the sugar in there. One of the products of fermentation is acid. So our bacteria ferments the sugar, make acid, turns the media yellow. Now we can tell the difference between things that fermented it and didn't ferment it. That's differential. So it's just telling difference, and it's always based on color change relating to some biochemical aspect of the organism. Okay. All right. So we're going to stop there with the Kahoot and really quick go through some of the questions on our. We'll get through all of it on the normal days. I just had to take an hour going over the course today. So this is our lecture activity exercise for today. Chapter 3. This is the case study. All right. And we have to identify where in the case study each of the 5 I's are. So I'll put another note up. I won't always put the notes up, but today we need them. First I was inoculation. Put the sample on some media to grow it. Next one is incubation. Grow sample at ideal conditions. Next is isolation. Separate the pathogen from contamination. Next is inspection. If you're inspecting, this is doing tests. And identification, using the test results to identify the organism. Not everything has a PCR test. Get that out of your head. Not everything has a PCR test. And if you're doing a PCR test, you have to know what you're testing for. And what I mean is, you can't say, oh, my patient, they have some sort of respiratory infection. Let's just run a PCR. No, that's not how it works. You have to run a PCR to test for COVID. You have to run a PCR that tests for flu. You have to run a PCR that tests for RSV. There are PCRs that can test for more than one thing, but they're very rare. So you have to know what you're testing for, and you have to have the tools in the PCR machine that test for the thing. And that gets complicated, and we'll learn about that in chapter 6 and 8. So don't worry about the details of it right now. Chapter 6 and 8 in unit 2, we'll go over that stuff. But PCR, just to be clear, it's not a magic, what is this? And it tells you. You have to know what you're testing for, and you have to have like the genetic information in the machine to compare it to. So otherwise it can't tell you. So if your patient has a respiratory infection and you're testing for COVID, it'll just tell you if you have COVID or not. it's not going to tell you that they have whooping cough. Instead, if you're just testing for COVID, it doesn't tell you that you have flu or RSV. It just tells you if you're testing for COVID, if you're positive or negative for COVID. That's all it'll tell you. So you have to test each thing individually. So if your patient comes to you with a less common disease, like anthrax or something, you're generally not going to be able to test for that on PCR. So all of the other testing methods might be used, like our selective and differential media and things like that. Test for the presence of these things, okay? So it's important to keep in mind, not everything has a PCR, or in some cases, there's no need to use the PCR. I also want to remind you, the PCR is expensive. It's more expensive than growing some stuff on a special media. So if you think somebody has staph, why would you do a PCR when you can just grow it on this special media that's designed to grow staph? It's way cheaper for your patient and just as reliable. Yes.
So I worked at a vet clinic. At first staff, we worked with bulldogs a lot. And we'd always just do a cytology.
Things like that, just looking at the cells under the microscope can often tell you more about it. And so when you look at the microscope at a bulldog's ear sample, and you see a lot of nucleated big cells that are kind of budding off one another, you're like, this is yeast. This is yeast. And it's more likely going to be certain kinds of yeast in those dogs. That's going to, you know that, and that's enough to tell you how to treat the thing. You don't need to run PCR that's more expensive. Definitely in veterinary medicine, that's an area where you are way more constrained because the people can't afford all those tests because they don't have insurance for their pets. You get what I'm saying, right? So it's a little bigger problem for vet than it is for nursing. All right, so we'll go through some of this. 5-year-old Liam brought to Yost Memorial Hospital. The kid had crusted lesions around his face and mouth, okay? When we asked the mom, it started out as small red spots that developed into honey-colored crusted sores over the course of a few days. Doesn't seem to have any fever or systemic signs. Other students at his preschool are also showing this crusty yellow stuff on their face. So we think this is probably impetigo. Anybody ever heard of that? You know, so it's extremely common in kids. I want to be very clear on this. They get that little crustiness on their face. This is impetigo. It's caused by either Staphylococcus aureus or Streptococcus pyogenes. Also common causes of a lot of things, So impetigo is one of these. All right, so we did a swab to collect the sample of the crusted lesions. We put it onto different media. Blood agar, mannitol salts agar, and McConkey agar. The blood agar looks to see if we're able to break down red blood cells. That tells us about the bacteria. The mannitol salts agar favors the growth of gram-positive organisms. If it turns that media yellow, it's Staph aureus. And I'm telling you that in this so that you also are able to follow what's going on with the media. McConkey agar is used to look for gram-negative. Gram-negative bacteria will grow on that. And I know you don't know what a gram-positive or gram-negative is. That's chapter 4 stuff. Don't worry about it yet. We get in there. Okay, we get in there, it'll happen. Have the faith. Okay. So anyways, so when we put his swab onto those media, that's the inoculation step. We've got inoculation out of the way. Then we took those plates and grew them for 48 hours at 37 degrees Celsius. That's body temperature, 98.6 degrees Fahrenheit. So that is our incubation phase. Then we look to see what grew on what and things like that. That's already, we're getting into the inspection phase. We skipped a little bit of isolation. We'll kind of skipped it. Except in this next part, right? To support our preliminary diagnosis, it grew on this, but not on this. So we think it's probably staph. But to inspect that further, we took one isolated colony on mannitol salts agar and did a quad stream, which you're going to learn about in lab. It's a way to separate out the cells and make sure that you've purified your culture. We say, oh, there's a ton of golden yellow colonies. We'll take one of those separated colonies, spread it out just to make sure there's no other contaminants in it. When we get the cells spread out individually, each cell goes up into a colony. You guys have an idea of what a colony is. It's those little spots that grow up on the media. They come from one cell multiplying on top of itself. So if you take a sample of that colony, it should be just one cell that multiplies, just a copy of that one cell. Make a new culture with that. In sterile media, you've got a pure culture of that organism. That is the isolation step. So we did that after we kind of got some results on our media. Once we isolated that media, they did PCR. You see how it says PCR. We're going to do real-time PCR. If the organism that we're doing PCR for strep, it's just going to be positive for the strep. or negative for the strep. If we do PCR for staff, positive for the staff or negative for the staff. So if you're looking for Staph aureus genes and it was positive for the PCR, it had Staph aureus genes in it. So that's another tick for our inspection. So now it grew on mannitol salt agar, it turned it yellow. It broke down red blood cells. It didn't grow on McConkey agar, and it had a positive PCR. That all supports it being Staph aureus. That's the identification part. Okay, we track that? All right, that's it. So we have lab at 11, right? Take a minute, get a breather, get a drink. Don't bring drinks into the lab. There's a little table outside of the lab where you can set your drinks. You're welcome to leave at any time from the lab to go to the bathroom, take a drink outside, but just don't keep a drink in the lab. If you don't have your lab coat today, you're okay, but do have it for Thursday, okay? Do have it for Thursday. I recommend either buying it from Walmart or Amazon. If you get it ordered on Amazon that you don't have it by Thursday, just let me know, okay? But show me evidence that you ordered it. If you did order it from Amazon, you don't have it, right? You get what I'm saying? Yeah. Huh?
What room number, I'm sorry?
So room number 200A is upstairs. At the top of the stairs, turn right, and it's like around the corner.